theories of enzyme activity. In today's session, we will deal with the same. Entropic effect. Enzymes enhance reaction rates by decreasing entropy. When correctly positioned and bound on the enzyme surface, the substrate are strained to the transition state. This is referred to as proximity effect. Chemical reactions need physical opposition of two reactants. The occurrence of collision between two substrate molecules is determined by statistical probability. Since enzymes usually are present in low concentrations, the collision probability is less and hence the reaction velocity is low. But a complex formation between the enzyme and the two substrate molecules can improve the collision probabilities manifold. This is called rapid rate of reaction. In this diagram, we can see that substrate bind to the enzyme at the active site, forming enzyme substrate complex. Next is product substrate orientation theory. Enzyme has appropriate three-dimensional structure to keep the specific orientation so that the reaction groups come into physical opposition leading to speedy reactions. It has been shown that hydroxyl groups of the sixth carbon atom of glucose and the terminal phosphate group of ATP are juxtaposed with the help of hexokinase. In this diagram, we can see glucose is converted to glucose 6 phosphate in the presence of enzyme hexokinases and conversion of ATP to ATP. In 1913, Michaelis Menten theory. In 1913, Michaelis and Menten put forward the enzyme substrate complex theory. Accordingly, the enzyme combines with the substrate to form enzyme substrate complex, which immediately breaks down to the enzyme and product. E plus S gives rise to ES complex, further leading to E plus P or enzyme plus product. In this diagram, we can see that enzyme plus substrate form enzyme substrate complex. The enzyme acts on the substrate, finally leading to enzyme product complex, and enzyme and products are released. Alkaline phosphatase hydrolyzes a number of phosphate esters, including glucose 6 phosphate. The active center of this enzyme contains a serine residue, and that step is taking place in two following steps. First, enzyme serine oxygen hydroxyl group plus glucose 6 phosphate gives enzyme serine oxygen phosphate plus glucose. And the next is enzyme substrate oxygen phosphate gives enzyme substrate hydroxyl plus Pi. Thus, the overall reaction is glucose 6 phosphate is, is equal to glucose plus Pi. In this reaction mixture, the enzyme substrate complex, that is E serine oxygen phosphate, has been isolated. If the concentration is increased, the forward reaction K1 is increased, and so K3 as well as the total velocity is correspondingly enhanced. Thus, three constants may be made into one. Km is equal to K2 plus K3 upon K1, where Km is called as Michaelis constant. It has been further shown that velocity is equal to Vmax S plus divided into uh, Km plus S. When the concentration of substrate is equal to Km, that is when S is equal to Km, the reaction becomes velocity is equal to Vmax into S upon S plus S, which is equal to Vmax into S upon 2S, which is equal to Vmax by 2. Fischer template theory. It states that dimensional structure, three-dimensional structure of the active site of the enzyme is complementary to the substrate. Thus, enzyme and substrate fit each other. Substrate fits on the enzymes, similar to lock and key. Lock can open by its own key only. In this diagram, we can see that substrate binds to the enzyme just as a key opens its lock. However, Fisher envisaged a rigid structure for enzyme, which could not explain the flexibility of shown by the enzymes. Here we can see that substrate fits into the fitly to the enzyme, but the substrate analog cannot fit into the enzyme. Conformational changes are occurring at the active site of enzymes, concomitant with the combination of enzyme with the substrate. At first, the substrate binds the specific part of the enzyme. This leads to more secondary binding and the conformation changes. The substrate induces conformation changes in the enzyme such that precise orientation of the catalytic groups is affected. In this diagram, we can see that amino acid side chains are arranged haphazardly, uh, forming unfolded protein with no activity. But when the folding process begins, the amino acid side chains from the active center of enzyme start building up, leading to folded protein with enzyme activity. A simplified explanation is that a glove is put on the hand. At first, the glove is in a partially folded position, but later, hand can enter into it. When the hand is introduced, the glove is further opened. Similarly, conformation changes occur in the enzyme when the substrate is fixed. When the substrate analog is fixed to the enzyme, some structural alterations may occur, but reaction does not take place due to the lack of proper alignment. Allosteric regulation can also be explained by the hypothesis of co-strand. Coming to active center of enzyme. Catalysis occurs at the active center or active site. For example, active site of ribonuclease enzyme is considered. The catalytic site of ribonuclease also lies within the hydrophobic cleft, where on the opposite side of the binding site for uridylic acid, its amino acid at the active center are 6th and 41st position there is lysine present, whereas at the 12th and 119th position histidine is present. These active groups are brought to the specific orientation by the tertiary structure of the protein. Proteolytic enzymes having a serine residue at the active center are called serine protease. Example, pancreatic protease, coagulation factors. Here are certain examples. Serine protease contains trypsin, chymotrypsin, and clotting factor. Cysteine proteases contains example papain. Aspartyl proteases is example is renin and retroviral proteases. Carboxyl proteases is present in pepsin. Metalloproteases is present in carboxylpeptidase. Protease inhibitors used as drugs. Example ACE inhibitor. Example captopril. And HIV protease inhibitor. Example is ritonavir. Here is the important amino acid at the catalytic site. For chymotrypsin, it is histidine, asparagine, and serine. For trypsin and thrombin, there is serine and histidine. For phosphoglucomutase and alkaline phosphatase, there is serine present. For acetylcholine esterase, there is serine. For carbonic anhydrase, there is cysteine. For hexokinase, there is histidine. For hexokinase peptidase, carboxypeptidase, histidine, arginine, and tyrosine are present. For aldolase, lysine is present. At the region of the enzyme where substrate binding and catalysis occurs is referred to as active site or active center. Although all parts are required for keeping the exact three-dimensional structure of the enzyme, the reaction is taking place at the active site. The active site occupies only a small portion of the whole enzyme. Generally, active site is situated on a crevice or a cleft at the enzyme molecule. To the active site, the substrate is bound. The binding of substrate to the active site depends on the alignment of specific groups or atoms at the active site. During the binding, these groups they may realign themselves to produce the unique conformation orientation so as to promote exact fitting of substrate to the active site. The substrate binds to the enzyme at the active site by non-covalent bonds. These forces are hydrophobic in nature. Amino acid or groups that directly participate in making or breaking the bonds present at the active site are called catalytic residues or catalytic groups. This active site contains substrate binding site and catalytic site. Sometimes these two may be separate. Coming to the thermodynamics of the enzyme. From the standpoint of energy, the enzymatic reactions are divided into three types. First, exergonic or exothermic reaction. Here, energy is released from the reaction and therefore reaction essentially goes into completion. Example, urease enzyme, where urease gives ammonia plus carbon dioxide plus energy. At the equilibrium of this reaction, the substrate will be only 0.5% and product will be 99.5%. Such reactions are generally irreversible. Second part is isothermic reaction. When energy exchange is negligible and the reaction is easily reversible. Example, pyruvate plus 2H gives lactate. 3 is endergonic or endothermic reaction. Energy is consumed and an external energy is to be supplied for these reactions. In the body, this is usually accomplished by coupling and the by coupling the endergonic reaction with an exergonic reaction. Example, hexokinase catalyzes the following reaction: glucose plus ATP gives rise to glucose 6 phosphate plus ADP. Here ends our session. Kindly like, share, and subscribe the channel. Thank you.